So, as you may have heard, the exams are graded. They are not yet available. The people in the morning section, recitation, got them this morning. Our intention is primarily to have you pick up exams in your recitation section. However, there will be a graduate student over in the BB office, ALS 2011, handing out exams at 1 o'clock. We'll do that for a short period of time. But other than that time, you can either get your exam during the recitation or during the hours posted on the BB office in terms of when they're making exams available. So do not, underline, do not bug the BB office to get your exam at times other than that. Okay? So only during the times that the BB office is available, plus 1 o'clock today. We'll do that at 1 o'clock today. Okay? Okay. So we scrambled, and we being the graduate students, scrambled to get that exam to you by today. And I think they did a great job, and I think they deserve a lot of credit for the amount of effort that they put into getting that together for you. The average on the exam was 62, which was a little low. I will post a grade distribution, and I didn't get the grades entered in my own system until last night. So I don't have that grade distribution done for you yet. I will plan tentatively to post that this evening. A couple of you have asked about highlights. The highlights are behind. Yes, the highlights are behind, folks, but we've been doing a few things. So please be patient with us with respect to that. I don't forget that we've got highlights to give to you. I do post things for you every time, right? So please just be patient with that. That's important. Yes? Question? Yeah? ALS 2011. Yes? The grade distribution will be posted on the schedule page. Now, one of the things I've learned sort of indirectly is that a lot of people are finding the schedule page not by the bookmark that I sent out to everybody, but rather by Googling it. And that's fine if you get the right one, but if you Google Kevin Ahern's schedule page, you're going to get about 8 of them. And so I keep getting questions from students going, oh, the dates are all wrong. Well, if the dates are all wrong, you're looking at the wrong one. So tonight, when I announce the availability of the grade distribution, I'm going to once again send out the proper URL for you to link to look at the schedule page. Please bookmark that. Don't Google it, because you're going to find the wrong one and you're going to get wrong information, right? So make sure that you use the one that I send to you, okay? Okay, other comments, questions? I know I'm harping today, but it was a scramble to get it together, and as I said, the graduate students really did a wonderful job with that. Okay, well, we turn our attention now to what will be the last that we're going to talk directly about enzymes, and that's having to do with enzyme regulation. When we talk about enzymes in the future, it'll be about individual enzymes, not enzymes as a whole. So this last section, this last segment on enzymes is a very important one. I think I've used the analogy in this class about an enzyme being like a race car, and that enzymes are speeding reactions so much that we could imagine that if we speed it too much, things may get out of control before we get it slowed down or stopped. And cells can't afford that, because when cells break down molecules for energy, for example, it takes more energy to build them back up than they get out of what they break down, which means it's a losing proposition if cells break molecules down and then turn around and just remake them because they went too far. Okay? So it's important then that cells have controls on the molecules or on the enzymes that are breaking down and making molecules. Similarly, just like we don't want to be breaking things down when we've got plenty of energy, we also don't want to be making molecules when we don't have sufficient energy. Because when cells don't have sufficient energy, they should be using that energy ultimately to staying alive. And if they're wasting energy in terms of making molecules that they don't otherwise need, then that also would not be a good idea. So enzyme regulation is very important. We need to understand enzyme regulation in order to understand the process of metabolism. And we're moving very much in the direction now of metabolism. Metabolism, of course, being the biochemical reactions that occur in cells. Well, let's take a look at some of the mechanisms that cells use to control enzymes. What you see on the screen is a beautiful picture of the enzyme hexokinase that I talk about all the time. And hexokinase catalyzes a reaction involved in sugar metabolism. And the enzyme is shown in that big blob. There's two big blobs in the middle. And the substrates that they work on are shown above and to the right. And I like this because it's shown approximately to scale. So you can get an idea about the immensity of enzymes relative to the molecules that they work on. Okay? Well, what are the mechanisms that cells use to control things? I've already talked about one of them already, and that's allosterism. I'm going to remind you that allosterism is a means of controlling enzymes in which binding of a small molecule to an enzyme affects the enzyme's activity. Allosterism is a means of controlling enzymes where binding of a small molecule to an enzyme helps to control its activity. Now, I've talked about allosterism and reminded people that under allosteric control, we typically do not have on-off switches, but rather we have volume control. We'll see a little bit of that later today. Increasing the volume or decreasing the volume, but not having a simple on or off switch. Allosterism works in terms of binding of small molecules. We'll see some different categories of those. And what we will see about allosterism is that allosterism can both activate and inactivate enzymes. So there's no given that it's going to be active or inactive. A given effector, and by the way, an effector is something that one of the molecules that binds to the enzyme. A given effector will always act in the same way. Okay? We're going to talk later today about the enzyme known as ATCase. Yes? I'm sorry? I haven't said anything about concentration yet, no. Okay? We'll see some examples of that later. Yes. But concentration will affect this, yes. Okay? That ATCase, for example, when it binds to ATP, gets activated, and when ATCase binds to CTP, it gets inactivated, or the volume's turned down versus the volume being turned up. Okay? All right. ATP always affects the enzyme in the same way. CTP always affects the enzyme in the same way. Okay. A second way of controlling enzymes is by covalent modification. Now, covalent modification occurs when we make or break covalent bonds. We will see both examples. We will see both examples today, actually. Okay? So covalent modification involves the making or breaking of covalent bonds. Usually, when we break a peptide bond, which is one of the kinds of bonds that get broken, usually that has the effect of activating the enzyme. Okay? Not always. The other common type of covalent modification that we will see is phosphorylation or dephosphorylation. And they have totally unpredictable effects, but they're consistent for the same enzyme. Okay, the third means of control of enzymes is by the control of the synthesis of the enzyme. Later in the term, we will talk about the pathway known as gluconeogenesis, that is, the pathway by which our cells or our body makes glucose. It turns out that we don't make glucose in all the cells of our body. Our liver is one of our major organs of the body for making glucose, and that's because the enzymes necessary for gluconeogenesis are synthesized there. If I look at my skin cell, 2 of the critical enzymes necessary for making glucose are absent. And if they're absent, there's no way of making glucose in those cells. So we can think of this, excuse me, just one second. If we think of this, then that is a perfect on-off switch. If the enzyme is not being made, it's off, right? Question? So is glucose more favorable to be made in the liver versus the muscles? Glucose is not made in the muscles. So it's absolutely. We have three primary places in the body where glucose is made. You don't need to know this right now, but liver, kidney, and in the testes. That's the primary places where we see glucose being made. It's not made in skin cells, it's not made in muscle cells, it's not made in brain cells. And those impose certain limitations on those cells, as we will see. The body has some ways to help feed those cells, which is important. Okay, so control of synthesis of the enzyme, I should have added of the enzyme to that. Control of synthesis of the enzyme is a very good way of controlling the enzyme's activity. And the fourth one that's there is the availability of substrate. If cells don't have substrate available, of course, there's no way for the enzyme to be active. And that actually can be modified just a little bit. The little bit is that if we think about things like Km, the affinity of the enzyme for substrate, if an enzyme has a high Km for substrate, it's going to take a lot of substrate before that enzyme is active. And that's actually used in some control of metabolic pathways. Yes, question? A question? Have a question? Yes? I'm sorry, could you repeat the question? Yes. So the question is if you're breaking a peptide bond and activating an enzyme, is that inconsistent because you're changing the structure of the enzyme? Well, the answer is no, it's not. So most of the enzymes that are activated by breaking of peptide bonds require the breaking of the bond to open up the binding site for the substrate. So in the absence of that, there's no way for the enzyme to bind the substrate. When you cleave that bond, now the substrate can bind. That's one example, and there are others. Okay. But yes, it does change the enzyme. No question about that. All right? But we can change an enzyme from being inactive to being active, and that's what's happening here. Okay? Good question. Okay. All right. So we've all seen this. All right? This is a typical V versus S plot. We know that if we have an enzyme that does not act allosterically, that we see a plot very much like what you see on the screen. It's hyperbolic in nature. We get a Vmax, we get a Km, and bang. Allosteric enzymes, and I want to qualify this, certain allosteric enzymes will behave in the manner that you see on the right. It gives a sigmoidal plot. That's that S-shaped curve. And when you see something like that thing on the right, it tells us that the substrate is affecting the enzyme by its binding. We saw the similar thing when hemoglobin was affected by its binding to oxygen because it favored the binding of additional oxygen, and so that binding of that initial one changed the way that hemoglobin was. The same thing happens when a substrate is an allosteric effector. We're going to see there are things besides substrates that are allosteric effectors. They won't exhibit the phenomena that we see here. Okay. Doesn't change it, does change it. Okay? Now, this means that in order for you to understand what's happening with allosteric effects, that you have to understand different types of effectors. Now, I've already described one. That is where the substrate is an allosteric effector. We'll see in the case of ATCase that aspartate, which is a substrate for the enzyme, is an allosteric effector of the enzyme as well. The more aspartate you put in there, the more the enzyme changes, and it's changing. When you activate an enzyme, you're changing the enzyme from the T state into the R state. Similarly, if I add an allosteric effector that reduces the activity of the enzyme, I'm changing it from the R state into the T state. Okay, well anyway, you can see the effects here. First, I've shown some, I think I backed, did I skip up? I thought I had, there, homotropic, okay. So, first I want to describe homotropic effectors, all right? Now, homotropic effectors are the type that I've been describing to you. That is, they are substrates of the enzyme. Aspartate with ATCase is a homotropic effector because it's both a substrate and it affects the structure of the enzyme, thus affecting its activity, changing it into the R state. A heterotropic effector, on the other hand, is something that is not a substrate for the enzyme, but also affects the enzyme. Right? Now, anybody see any parallels to different types of inhibition that we talked about? What were the two types of inhibition we talked about in class? Competitive and non-competitive, right? And how did competitive inhibition manifest itself? Binding to the active site, right? Now, this is not an inhibition in the case of aspartate. It's a substrate, but it is occurring at the active site, right? If I have a heterotropic effector, it models non-competitive inhibition because it's not like the substrate. It's not binding at the active site, but it's binding elsewhere and affecting the enzyme. It can be inhibitory, as we will see. It can also be stimulatory, as we will see. Okay? But in the case of heterotropic effectors, they are binding to sites on the enzyme that are not the active site. Very important consideration. We'll see an example in just a bit. Okay. Well, here's the enzyme I've been talking about. It's known as ATCase, or if you want the full mouthful name, it's aspartate transcarbamoylase. I'll take ATCase. Okay? ATCase is a complicated enzyme, at least to glance at it, but in fact it's relatively simple. We will see that it contains two types of subunits. It has 6 subunits known as regulatory subunits, and it has 6 subunits known as catalytic subunits. The 6 catalytic subunits are all the same. The 6 regulatory subunits are all the same. Now you might say, what are the green things there? The green things aren't actually a part of the enzyme. They're just there to show the symmetry of the enzyme. Okay? So there's 12 subunits to the enzyme, 6 regulatory and 6 catalytic. The reaction catalyzed by ATCase is a little complicated, but you can see it starting with bicarbonate at the top and finishing with carbamoyl phosphate at the bottom. Okay? This enzyme requires input of nitrogen, it requires input of phosphate, it requires input of energy, and the upshot is that a small molecule containing phosphate, carbon, and nitrogen is made, and that intermediate is necessary to synthesize pyrimidine nucleotides. Okay? So this is what we call the first step in the synthesis of pyrimidine nucleotides. Pyrimidine nucleotides being CTP, UTP, and ultimately DTTP. Okay? This step is the first step in that process. Now you're going to see in a little bit that it's the first step. There are many steps in making pyrimidine nucleotides. We will be interested in the end product of that pathway because we will see that it will affect this enzyme. Okay? All right. Now, aspartate comes into this, as you will see in a reaction in a minute, which is actually bringing in some of the nitrogen that's there. Here's another view of what the enzyme is doing. Okay? Another view. Carbamoyl phosphate is, from that last set of reactions, is combining with aspartate to make this intermediate, carbamoyl aspartate. Okay? This molecule, even though we talk about it being the first step in the process, is, as you've seen from the last one, actually the second step in the process, right? We had to make that carbamoyl phosphate in order to make this guy. This is the reaction that carbamoyl phosphate, I think I may have said that earlier incorrectly, that ATCase is actually catalyzing. Okay? This reaction is what ATCase is catalyzing. We can see the addition of aspartate. Aspartate is a substrate, and that substrate is necessary for the reaction to occur. Now, what is aspartate? It's an amino acid, right? Yes, it's an amino acid, Kevin. Okay. Aspartate is an amino acid. Why do cells want to make Nucleotides. It's an easy question. DNA replication, why do cells want to replicate their DNA? They want to replicate themselves, right? Cells like to make whoopee. Right? And if you don't have the goods, you ain't going to make whoopee. Right? Snicker, snicker, snicker. Okay? So, when is a cell going to want to make whoopee? Not after a couple of beers, although that might help too. A cell wants to make whoopee when it's time to make whoopee, right? And how does a cell know when it's time to make whoopee? What would a cell need to know that, hey, you know, I got some free time. I got some XYZ. I'm going to make some whoopee. What is a cell going to ask the question of to be able to do that? What does it need to know? You said it needs to make DNA, right? What does it take to make DNA? Nucleotides. What does it take to make nucleotides? What does it take to make anything, folks? Energy. Right? If I don't have the energy, oh, not tonight, dear. Right? Not tonight. I've had a terrible day. I have no energy, right? Cells have to have energy. What else do cells need besides energy and the desire to make whoopee? Materials. What kind of materials? Come on, guys. I hate pulling it out of you. ATP is an energy indicator, but what do they need besides energy? We've been talking about them for the past 2 weeks. They have amino acids in them. Proteins, right? Okay. They have to make proteins. It takes energy to make nucleotides. It takes energy to make proteins. And what does it take to make proteins? I'm going home. How about amino acids, okay? Can we just compromise on amino acids? Would that be good, right? They need amino acids. And do you see an amino acid on the screen? Yes, Kevin, we see an amino acid on the screen, right? Okay. Right, thank you. All right. Yeah, hallelujah, right? I see an amino acid on the screen. So this amino acid might be an indicator that a cell is using that, hey, we've got enough amino acids to make whoopee, right? Make sense? It's not going to tell everyone, but if I've got enough of this one, maybe I've got enough to go ahead and do my thing. We're going to see some other things that indicate things to cells. That's why ATCase is so important. ATCase catalyzes this reaction. It's affected by the amount of aspartate. If cells have very little aspartate, well, we're not ready to go have a wild Friday night, guys, right? Or thirsty Thursday, as the case may be. We're not going to have that, right? If we have plenty of aspartate, okay, this system is go. We don't have all systems yet, but this system is go. All right, so aspartate is an amino acid. This enzyme is affected by ATP. I've already given that secret away. ATP binds as a heterotropic effector and activate or inactivate. It's going to activate, right? Because, again, it's indicating I have high energy. If there's plenty of ATP, you want this enzyme to be active because, hey, you want to be active, guy, right? Right? CTP. CTP is also an allosteric effector. It's also a heterotropic allosteric effector. It's the end product of the pathway that this reaction is the first/second of. Okay? What does that mean? Well, if cells have plenty of CTP, do they want to make more? No. So this is indicating, hey, you got plenty of pyrimidines, dude. It's time to start making some purines. Stop making pyrimidines. This enzyme is taking the pulse of the cell. It's taking the pulse. It's got an indication of amino acids. It's got an indication of energy. and it's got an indication of pyrimidines. That's pretty cool. One enzyme, all three of those things built into it. Okay? Now, we'll see that there's even more efficiency built into it than what I've said in just a bit. All right? So, there are the substrates, and there is the plot of V versus S for ATCase as a function of aspartate concentration. And no surprise, it has the plot that we would expect to see for a homotropic effector. Aspartate is a homotropic effector because it's a substrate and it's affecting the enzyme. Okay? Why does it say heterotropic effector? It should say it's a homotropic effector. We shall fix that. Okay. It is a homotropic effector, not a heterotropic effector. Okay. The binding of aspartate by ATCase favors the R state. It favors the R state, meaning that the enzyme volume is being turned up, meaning that when amino acids are plentiful, we're about ready to go and do our thing. Okay. What is the effect of ATP on this enzyme? If we look at the blue line, we see the enzyme as a function of aspartate concentration in the absence of ATP, and the red line shows the activity of the enzyme in the presence of ATP. We can see that the reactivity is shifted slightly to the right. What does that mean? It means the following. If we draw a straight line from one aspartate concentration and we draw it upwards, what do we see? We see that in the presence of ATP, the V sub 0, that is the velocity of the reaction, is increased at the same concentration of aspartate. The enzyme's volume has been turned up. The enzyme is activated. Okay? Compare no ATP to ATP. If we look at the activity of the enzyme as a function of CTP concentration, now notice in this last one we were looking at aspartate concentration on the x-axis. In this reaction on the next slide we are looking at CTP concentration on the x-axis. We're still measuring the velocity of the reaction, which means we've got a constant amount of aspartate and varying amounts of CTP. As we increase the CTP concentration, we see that the velocity of the reaction decreases, telling us very clearly that CTP is inhibiting the enzyme. Okay? Notice, CTP is not a substrate, guys. It's a heterotropic effector. It's binding elsewhere on the enzyme than the active site. Okay? CTP is turning the volume of the enzyme down. It's converting it from the R state to the T state. ATP was converting it from the T state into the R state. Okay? Now, here are the different sites on the enzyme. So, to refresh what I've just said, aspartate is a substrate, but neither ATP nor CTP is, and all three affect the enzyme. Okay? Question? Oops, somebody just hiccuping. Okay. All right. 6 regulatory subunits. They're labeled as R1 through R6. Okay? ATP and CTP are binding to the regulatory subunits. Those are the non-catalytic subunits because they don't need to bind to the active site. Okay? ATP favors the R state, CTP favors the T state. And finally, we have 6 catalytic subunits, C1 to C6. And C1 to C6 are, each of them has an active site. Okay? Everybody with me? Okay. So, aspartate binds to the catalytic subunits and favors the R state. Okay? All right. Allosteric enzyme kinetics, you've seen this a couple times now. At low substrate concentration, ATCase is in the T state. As the substrate concentration increases, ATCase is increasingly in the R state. And at high, ATCase is mostly in the R state. Very much like what we saw with hemoglobin binding oxygen and changing its affinity for oxygen as a function of oxygen concentration. Okay. So, it's most active when energy is high. And this figure is probably a bad one to show this on because ATP is not affecting the S shape of the curve directly here. Okay? And it's least active when the pyrimidine concentration is high. I just simply use those high and low points here to show you that the enzyme is active or inactive with each effector. Okay, So now I promised I was going to talk about the overall pathway that this enzyme was participating in. Okay? We see carbamoyl phosphate being converted into carbamoyl aspartate in the reaction catalyzed by ATCase. And whether it's the first or the second reaction for our purposes doesn't really matter. But what does matter are those arrows that follow after the carbamoyl phosphate. Those arrows indicate that there are other reactions, unshown, other reactions that are necessary to convert carbamoyl aspartate ultimately into CTP. I told you on a previous slide that CTP was the end product of the pathway, and you know that CTP is a pyrimidine. Okay? So we can see this overall process occurring on the screen, right? All right? As CTP accumulates, it starts binding to ATCase and starts to inhibit the enzyme. Okay? Cells, on the other hand, cells in a high energy state with a lot of ATP have ATP binding to the enzyme and activating it. Who's going to win? ATP or CTP? What do you think? What's going to determine who wins? Concentration. What else? Now that you people are enzyme experts. What's the other consideration with binding? Affinity, and what's affinity measured by? Km, right? So Km and concentration are going to ultimately determine who wins. Right? Everybody with that? Now this turns out to be important because ATP, pyrimidine or purine? Purine. In DNA, what do pyrimidines pair with? Purine. If I have too much pyrimidines, what do I want to make more of? Purine, right? So, if I have too much pyrimidines, the first thing I've got to do is shut this guy off, right? If I have too many purines, what do I need to make more of? Pyrimidines. And who's turning this enzyme on? A purine. We actually see the evidence of complementarity right here in this enzyme. Purines favoring pyrimidines, pyrimidines inhibiting themselves. Pretty cool. Okay? All happening in one enzyme. All happening in one enzyme. Okay? And the last consideration, of course, are amino acids. Amino acids, when cells have plenty of amino acids, they can make plenty of proteins, and they're ready to go to the next phase or take their relationship to the next level. That didn't get any laugh at all. Okay. All right. So I hope I have convinced you that allosterism is an important and powerful mechanism. Allosterism, as I said, does not generally involve an on or off switch. So there must be other ways that cells have of controlling enzymes, and there are other ways, as we shall see. Okay? Covalent modification is a very powerful and a very important way of controlling enzymes. All right? What you see on the screen is a set of reactions by which our body converts proteases from an inactive form to an active form. Now, proteases are things that are potentially really problematic for the body. We make proteases mostly in our pancreas, okay? At least digestive proteases in our pancreas, and they get released by the pancreas in an inactive form. And there's a very good reason for that. Because what do proteases do? They break down proteins, and our cells have a lot of proteins on their cell surface, and what do you suppose would happen if the proteases started attacking the cells that made them? You see problem? Definite problem, okay? If you have that problem, you develop a condition known as pancreatitis. Anybody know anybody with pancreatitis? Not uncommon. Okay. Pancreatitis is very painful. Pancreatitis involves literally what I just described to you. Proteases attacking the pancreas, and if it's not properly treated, it can be fatal. Okay? So the pancreas releases proteases in an inactive form, That inactive form has a name. They're called zymogens, Z-Y-M-O-G-E-N. Zymogens are inactive forms of enzymes. Here's an on-off switch, guys. They're released completely inactive. And in response to the question he had up here, one of the ways in which they're controlled and activated is by cutting specific peptide bonds that allow access of the substrate to the active site. In the absence of that, it's completely covered, and the substrate can't get there. Well, if the substrate can't get there, the enzyme is dead in the water. This is an on-off switch. Okay? Well, how do they get activated? The way that they get activated is through the system that you see here. Trypsinogen, when you see the O-gen on the end of a name, it indicates it's a zymogen. Trypsinogen is an inactive form of the enzyme trypsin, a protease. Okay? That nomenclature is not consistently used. You frequently see pro in front of an enzyme name to indicate an inactive form. So you see pro-elastase that's converted into elastase. You see chymotrypsinogen, which is converted into chymotrypsin, right? So you see a variety of different names that are used. You see on the screen a hierarchy of activation. Hierarchies that I call cascades. Cascades turn out to be very, very powerful and rapid ways of affecting the activity of a large number of enzymes almost simultaneously. Now, in the lecture on Wednesday, I'm going to talk about blood clotting. Because when we think about something that we want to have happen rapidly, blood clotting is a prime example. We don't want to bleed to death, right? But we also only want it to come on when we need it. We don't want clots forming in the absence of cutting ourselves, because then we can form clots in our system and have stroke or heart attack, right? So that's not good. All right. So these cascading systems are powerful because the activation at one level, let's say we convert, enteropeptidase converts trypsinogen to trypsin, all right? Now I have trypsin active. At each level, I've got, let's say, one enteropeptidase that could easily activate 10,000 trypsinogens. Now I have 10,000 trypsins that can activate 10 million of the next level of enzyme. And the further I go down, the more enzymes get activated. It's a cascading system that at each level increases enormously. With this, cell or the body can activate or inactivate a tremendous number of enzymes very, very quickly. In all the cases you see on the screen here, they're all activations. They're all activations here. Okay. Here's another system. I'm not going to take you through it. We'll talk about it later in the term, actually, but I just want to show you another system that's cascading in nature. This system is involved in signaling that we'll talk about later. And this signaling system involves the activation and or inactivation of selected enzymes in glycogen metabolism. And the same idea occurs. You don't need to worry about writing this all down because I'm just using it as an example. But the same thing occurs. We start at the top with a signal, in this case binding of a hormone to a cell surface receptor. Epinephrine or glucagon is a hormone. Okay? That activates a process that cascades down such that at the bottom of the pathway, we have turned on the breakdown of glycogen, and we've turned off the synthesis of glycogen. And because of this pathway, we can release a tremendous amount of glucose into our bloodstream so that we can escape danger. Okay? This cascading system works really, really well to turn on a lot of things at once or turn off a lot of things at once. And there are things here that are turned off. The glycogen synthesis is turned off as the glycogen breakdown is turned on. Okay. Well, let's look at that system again I showed you earlier in a little bit more detail. There's the enteropeptidase converting trypsinogen into trypsin. That is an activation of trypsin that's occurring. Trypsin, in turn, converts the chymotrypsinogen into chymotrypsin. It also converts proelastase into elastase. It converts procarboxypeptidase into carboxypeptidase. And it converts, the last one is not a protease, by the way. The last one is an enzyme for breaking down fat. It converts prolipase into lipase. In each case, you see the brown inactive form of the enzyme being converted into the green active form of the enzyme. Okay? Now, why would it turn on the breakdown of fat? Well, these things are occurring in the digestive system, and we do eat things besides meat with protein, right? We're eating things besides protein. We get a fair amount of fat in our diet. So in addition to breaking down the proteins into amino acids, we want to break down fats into fatty acids and glycerol, and that's what the prolipase is doing. The cascading effects are moving from left to right. That signal is increasing as we start with one enteropeptidase on the left and moving in the direction of the right. Yes, question right here. Trypsin is doing all of those things that are there. So the activation of trypsin is a pretty critical step in the process. We will see that chymotrypsinogen has an effect on itself, as we'll see in just a minute. Okay? These are complicated. Yes, question. What? Say it again. Okay, so the question is, what if you only need one of those four? In digestion, you want to throw as many things at a problem as you can. So in essence, it's a good question, but it's not a specific thing that the digestive system is wanting to do. Okay? And along that line, you just reminded me of something else I wanted to say. You think about, well, if you release all these enzymes that break down proteins, isn't it going to affect other cells? Right? You don't want to attack your pancreas. What about the rest of your body? Well, the place where these enzymes are active are in your digestive system. And the most rapidly proliferating cells in your body are in your digestive system. When you interrupt the replenishment of digestive system cells, that's when you get things like ulcers and problems. Okay? So it's important to understand that. When you think about aspirin, some people are very sensitive to aspirin, right? They get stomach problems with aspirin. Why does that happen? It isn't because the aspirin has acid in it, which is what people used to think. And I said, it's like, what bullshit is that, right? Okay? This little tablet has got more acid than my stomach has got in it? I mean, excuse me? No. Aspirin inhibits the synthesis of prostaglandins, and one of the prostaglandins is involved in stimulating the synthesis of digestive tissue. Some people are sensitive to it, some are not. I take aspirin, it doesn't affect me, but some people are affected by that. Okay. Well, I said that chymotrypsinogen it plays a role in its own activation. So let's look at how that happens. This is kind of an interesting process. You see the inactive form of chymotrypsinogen above, and you see the action of trypsin. Trypsin converts chymotrypsinogen, which is in the inactive form, to something called pychymotrypsin. Pychymotrypsin, right? It stimulates, all right, the production of pychymotrypsin. Pychymotrypsin is what we call partly active. Partly active pychymotrypsin will act on pychymotrypsin. And when it acts on pychymotrypsin, it converts pychymotrypsin into alpha-chymotrypsin. And alpha-chymotrypsin is the fully active form. Now, I don't care if you know which amino acids are affected, all right? I don't care if you know that. But you can see what's happening here. You see the breaking of a specific peptide bond near the amino terminus of chymotrypsinogen. And that disulfide bond holds the piece, keeps it from flying away. Okay? We also see, to make this guy fully active, we have to break out a couple of amino acids near the middle. We see that we lose amino acids 147 and 148 because two breaks happen in that enzyme at that point. Okay? And what's happening in this process is the active site of the enzyme is becoming fully exposed so that the substrate can bind. Now, one of the things about digestive enzymes, like proteases, is that they are self-destroying. Will proteases break down other proteases? Absolutely. Will alpha-chymotrypsin break down another alpha-chymotrypsin? Absolutely. So eventually, they just get all wiped out. If you let them go long enough in a tube, you'll ultimately have one left because they will all eat each other, basically. Talk about cannibalism. Okay. Okay. So there are some common protease precursors that are synthesized in an inactive form. I won't list them all here. I'm not asking you to memorize the list. But I just show them to you so that you see or hear, when you hear about these, you will know about them. You've heard of elastase already. I'll talk about blood clotting proteins next time. And the blood clotting proteins are a beautiful and complicated system of cascading reactions. Trypsinogen, I've talked about. Pepsinogen. Pepsin is the enzyme I mentioned that was active in the high pH, the low pH of the stomach. Okay, and other ones that are also protease precursors that are not involved in digestion. Plasminogen, for example, is involved in the breakdown. Plasmin is involved in the breakdown of blood clots. Okay. How do these things happen? Well, there's a variety of covalent modifications that can occur. I've mentioned phosphorylation. That epinephrine system that I showed you is largely a phosphorylation, dephosphorylation mechanism. Acetylation, adding of acetyl groups. Formylation, adding of formyl groups. Okay? Acylation, adding an acyl group, like a fatty acid, to an enzyme. ADP ribosylation, adding an ADP to a protein to affect its activity. Prenylation, adding a prenyl group. Okay, we'll talk about prenyl groups later. I won't define that here. Sulfation, adding sulfates. Ubiquitination. You've already seen ubiquitination. How does ubiquitination affect proteins? Favors their breakdown, right? So they're going to the proteasome. Okay? Gamma carboxylation. We'll see this one's very important in the process of blood clotting. Okay? Now, I want to focus for a minute on that last one, which is that gamma carboxylation. Gamma carboxylation we'll talk about next time, but it's actually modifying the side chain of glutamates. You see the side chain of a glutamate that might be found in a blood clotting protein. Okay? One of the things blood clotting proteins have to do is they have to bind calcium. Okay? And when we look at the reaction that's happening here, a carboxyl group has gotten added. Instead of the side chain of glutamate having one carboxyl group, it now has two. And with two carboxyl groups, it can bind to calcium. Pretty cool. Okay, how am I doing here? All right. Questions about that? You know what time I'm getting ready to say it is. It is time for a metabolic melody. The metabolic melody today is to a Christmas tune. It actually relates to what we talked about last time, not what I talked about today. So please join me in this one. I don't have a recording of it. To the tune of Winter Wonderland. Mechanism determines how an enzyme is working. Here are the ways that each elastase breaks a peptide bond so easily. Starting with the binding of the substrate. Catalytic triad is the star. Histidine's electron sync reacts to pull a protein from a serene's R. Then the alkoxide ion gets electrons a-flying. It makes a big fuss for one nucleus and breaks and makes a bond with carbonyl. Then the process switches in its action. Water comes to free the carbonyl. Loss of proton yields hydroxide ion. Attacking on the peptide bond, there's still what the enzyme releases. Otherwise, action ceases. The process is done until the S1 binds the substrate, starting up again. Okay, guys, see you.